colleague from KZN, anatomical pathologist, and speaking on molecular testing in breast cancer. Would you please come forward?
Good morning, everyone. So I'm going to be speaking to you about molecular testing in breast cancer, and I've chosen to do a very broad overview of the principles behind molecular testing in cancers in general, and then highlighting a few of the more specific areas in breast cancer to make it specific to this forum. Okay, so in understanding our testing of the background cellular changes in cancer, it's important that we go back to the hallmarks of cancer, which have been described by Hanahan. And these hallmarks have been expanded over the years to incorporate 10 different characteristics of tumors. So when you are considering a tumor in a patient and the cell biology of what essentially is a living entity, which is not playing by the rules, these are the characteristics which give it its malignant behavior. Okay, and most of you are aware of these, but I will, during my talk, try to show you how each of these are being examined to be targeted for different forms of therapy. So tumors, as we all know, based on their cell biology and physiology, evade growth suppressors. Okay, I will explain each one very briefly. The second one, and for the people online, I'm going in a clockwise direction. The second one is epigenetic reprogramming, which the actual DNA structure of the tumor cells with their chromatin and histone configuration actually influences how genes are expressed. The third one, which I will elaborate on a bit later, is the tumor cells learn to avoid our own immune destruction. Then the cells exhibit replicative immortality, and this has to do with telomere shortening, so they have found a way to evade that. Tumor cells also can produce their own inflammatory markers, like cytokines, which promote their own growth. One of the newer added concepts to the hallmarks of cancer is that of polymorphic microbes, which are still being researched. So this is the interactions between gut flora and microbes that are associated on the surface of our cells with the actual tumor cells and how they stimulate a form of inflammatory response which is synergistic to the growth of these cells. One of the newer hallmarks as well is phenotypic plasticity, where tumor cells lose their terminal differentiation and now have been described to trans-differentiate from one form of cell into another. And in doing that, they actually can stimulate their own regrowth. Okay. Angiogenesis, how oncologists will know about VEGF inhibitors. So tumor cells produce their own blood vessels and have the ability to invade others. The one newly described concept which is still emerging is that of senescent cells in the stroma. So these are inactive cells within the tumor microenvironment, and initially they were thought to be bystanders. Okay, this may be your stromal desmoplasia or your myofibroblasts, which are not part of the tumor, but recent studies have shown that these are not that inactive, and they are actually responsible for secreting growth stimulating factors. One of the other hallmarks of tumor cells is genomic instability. So I call this the bad photocopier syndrome. So tumor cells, as they replicate their DNA badly. So subsequent clones of tumor cells accumulate more and more mutations, and this leads to a lot of genomic instability and abnormal protein production. Okay? Tumor cells evade apoptosis, and they also dysregulate their own cellular metabolism, and one of these mechanisms is the alterations in aerobic glycolysis and how they actually metabolize glucose to proliferate, and that is also one of the new targets for anti-cancer treatment. Okay, and I will end using the sustaining proliferative signaling, which is one of the most common pathways. So the inherent pathways of cell growth, these tumor cells actually stimulate their own pathways, okay, and they become independent of normal stimuli. So when we are doing molecular testing, we are aiming to map the tumor cell physiology and microenvironment. And we do this to, as we all know, predict responses to treatment, to analyze the data and check whether it is prognostically significant, and also to identify possible types of intervention. What we look for is a broad range of markers which lead into those hallmarks. So we can sequence DNA using next generation sequencing. We can analyze mRNA, and we can also look for protein expression using immunohistochemistry. Okay, so I've just chosen a few very well-known instances in which we do sequencing. So everyone knows about the early breast cancer testing for the mRNA expression in it, and we have a few very well-known tests, and one of them is the Oncotype DX test. So if you have early breast cancer and it's hormone responsive, we send it for gene sequencing. We give you a risk recurrence score, and based on the risk recurrent and on the survival data of thousands of patients and archival material, we will be able to tell you your risk of recurrence and whether chemotherapy will be of benefit for the patient. And that is a widely used and accepted assay. Okay. So one of the other commonly described or well-known is that of tumor cells avoiding immune destruction. Okay, so this is the one that I have chosen to highlight. And I looked at the program today, and I see that the overview that I'm giving you today on the hallmarks of cancer feed into two other of our presentations. So you will know exactly where those two presentations fit in the hallmarks of cancer. So when it comes to immune checkpoints, okay, so in the early 90s, two scientists, James B. Allison, who is an immunologist, and Tasuku Honjo, who is a physician, did extensive research describing immune checkpoints in tumor cell physiology. Okay, and Allison described the CTLA-4 blockade pathway, and Honjo the PD-L1 pathway. And in 2011, the first approval for an immune checkpoint inhibitor was approved, or was given. And in 2018, these two scientists were awarded the Nobel Prize in Physiology and Medicine, okay, based on their work, because this has changed the way we view the medical treatment of cancer. Okay, so what were these two scientists describing? So basically, the PD-L1 pathway is a protective immune check pathway. It involves our own body's regulatory mechanism to not attack our own cells. So when you have one of our body cells interacting with a cytotoxic T lymphocyte, the T lymphocyte needs to know that the cell is not foreign and not abnormal. And the PD-L1 pathway, which is active, actually deactivates the cytotoxicity effects of that T cell. And this inactivation was extensively researched by the Japanese scientist, and he started manipulating this pathway during mice studies. And he discovered that once he was able to manipulate it, he could switch on the pathway and switch it off, and then began to wonder, can we use this mechanism to use our own T cell response to attack a tumor, okay? And that led to the development of the PD-L1 inhibitors. So on the upper part of the diagram is the tumor cell, the bottom part is your cytotoxic T lymphocyte, and you have the anti-PD-L1 drug, which is the red triangle, and that actually blocks the functioning of the anti-PD-L1 receptor and its ligand, okay? And many of us are aware of the checkpoint inhibitors that are involved in anti-PD-L1 blockade. So once we block this pathway, the cytotoxic T cell then is able to activate the pathway that results in tumor cell death. Now, this is a normal physiological response in our bodies. It actually occurs in virus-infected cells. So when you have a virus-infected cell, it expresses abnormal antigens. The cytotoxic T cell recognizes it and will destroy it using one of these pathways. Okay. So how do we test for this? So based on the clinical indications, I'm not a medical oncologist, so I'm not going to go into the treatment side of this and the side effects and the indications, but we will get a request for testing for PD-L1, and we run immunohistochemical studies on it. So this is an example of one of those tests. We look at protein expression, and the background blue staining are the nuclei of the cells. The brown reaction shows you the location of the PD-L1 protein, and in this particular slide, you have small round cells in the background, and you have those larger islands of brown cells, which are actually the tumor. And we evaluate the level of expression within the inflammatory cell component, excluding neutrophils, and on the membrane of the tumor cells, and we provide a score. And that score has been linked back to responses in tumors, and that score is then evaluated, and if it is positive within a certain range, then the patient may qualify for treatment. This scoring system that is used is tumor dependent, so I know I have one particular scoring system described there, but depending on the different type of cancers that you're doing from different sites, so whether it's bladder or lung or breast, in this case, we do different scoring systems. So that is utilizing PD-L1. Okay, so we are all the way back to the first slide. So I've chosen to keep this talk a little bit short today because each of the pathways that are described are described in numerous review papers, and I could have gone. Can I just go back one slide? Thank you. I could have gotten way into the pathways, and I think that it would have been just a little bit too confusing for this early in the morning. But we are now going to go and look at our hallmarks of cancer. And now I have added in the different categories of novel therapies which are being researched and show you where they fit into this pathway. So as you can see, the pathway that stimulates where the cells self-stimulate their own cellular proliferation is one of the most widely studied. Okay, and these are the categories of drugs that have been developed to block those pathways. One of the most well-known drugs that fall into that category is Herceptin. Okay, and we have all of these other drugs here. So Herceptin, much like the PD-L1, is evaluated utilizing immunohistochemistry, as well as fish studies if they are needed in the lab. And we use next generation sequencing to check if you have activating mutations in any of these pathways here. Similarly, later on you will hear about the cyclin-dependent kinase pathway, and the inhibitors that are being used in breast cancer. So this is a pathway that evades the normal suppressive. So all of our cells don't proliferate out of control because we have a check and balance system. So we have a system where we have an inhibitory mechanism that keeps our cells in check. And if that inhibitory mechanism is mutated, then your cells will be stimulated to proliferate, okay? So I've also talked about the immune checkpoint inhibitor. You know about that in much detail. So vaccine therapy is also a new form of therapy where subunits, for example. So one of the examples would be for HER2 positive tumors. So antigens from the HER2 protein are taken and incorporated into a vaccine and given to the patient to stimulate antibodies, which will attack the HER2 protein, which are overexpressed on the cells. So that's another form of manipulating the immune system to kill tumor cells. Okay, as I've mentioned before, aerobic glycolysis inhibitors are currently being investigated, but this is currently in the laboratories in vitro studies, and requires a lot of research from cell biologists into which pathways of ATP production are being activated in specific tumor cells before you can block them. And when it comes to genomic instability, we have the PARP inhibitor therapies, which have also been developed and come on board. And those inhibitor therapies are actually the subject of the next talk. So I've given you an introduction to an overview as to where all of these now fit into the hallmarks of cancer. So it can be quite daunting and complicated when you are approaching the different novel types of therapy, but if you use this as the framework and try to understand where it fits into, you should have a better understanding of it. And you will probably hear in the next talk that some of these therapies are actually synergistic and can be combined. So there are instances in which you can use immune checkpoint therapy in combination with the PARP inhibitors, okay? And that has currently been described with interesting results. So I thank you all for your time, and I hope that I have given you a slightly better understanding of the overall view of the therapies in breast cancer and the molecular underpinnings behind the development of these drugs. So I thank you for your time.